Nerve fibers function by transmitting signals quickly and over long distances. To facilitate this, some nerve axons are covered by a myelin sheath, which is produced by Schwann cells. The myelin acts as an insulator, resulting in a saltatory conduction, where the action potentials propagate between the myelinated sections of the axon, recharging at each node. This allows the transmission of a signal over long distances without degradation. The resting potential of an axon is -70 millivolts, with a neutral extracellular charge. Charge is maintained through the movement of charged molecules. Potassium, which has a relatively negative charge, exists inside the cell in high quantities, contributing to the charge difference. The resting charge is maintained through sodium-potassium pumps and leaky channels. While the sodium-potassium pump is active transport, moving into potassiums and removing 3 sodiums, and requires ATP, leaky channels rely on passive transports with molecules moving along their chemical and electrical gradients. Graded potentials are gradual changes in membrane potential as a result of ion channels. Graded potentials that make a soma more positive or negative make it more likely for the cell to experience an action potential. As neurotransmitters bind to pumps, sodium channels open, allowing the soma to become more positive, increasing relatively to the number of neurotransmitters. The threshold for an action potential is -50 millivolts. Once the cell reaches this level, an electrical impulse is sent down the cell. Depolarization makes the inside of the cell more positive, a result of excitatory neurotransmitters. Hyperpolarization makes the inside of the cell more negative, a result of inhibiting neurotransmitters. Voltage-gated channels open and close based on the internal charge of the cell, allowing for hyperpolarization and repolarization following an action potential. A threshold potential is the potential at which voltage-gated ion channels open, which is required for the propagation of action potentials. For depolarization, sodium channels open when the internal charge becomes -50 millivolts, while closing potassium channels and sodium-potassium pumps, which makes the cell increasingly positive. For repolarization, the voltage-gated potassium channels open once the charge becomes +30 millivolts, while the sodium channels close, and the sodium-potassium pumps remain closed, making the cell increasingly negative. Neurotransmitters are molecules that are used by neurons to transmit messages. After an action potential, the presynaptic cell releases neurotransmitters, which bridge the gap over the synaptic cleft to the postsynaptic cell. The postsynaptic neuron recovers the signal by detecting the neurotransmitters. The mechanism of synaptic transmission starts with an action potential that propagates from the axon to the synapse. Then, depolarization causes calcium channels to open, which allows calcium to diffuse into the cell. This signals vesicles containing neurotransmitters to merge with the cell membrane, which releases neurotransmitters into the synaptic cleft. Neurotransmitters diffuse across the synaptic cleft towards the postsynaptic cell. Then, finally, neurotransmitters bind to the receptors in the postsynaptic cell, which open sodium channels, causing depolarization and triggering an action potential. Acetylcholine, also known as ACH, is a neurotransmitter that is used in many synapses, including neurons and muscle fibers. After secretion, ACH diffuses to receptors on the postsynaptic cell, aiding in the creation of a graded potential in the cell. In a synaptic cleft, ACH is broken down into acetylcholine stearase, producing choline and acetate molecules. These products are reabsorbed by the presynaptic cell through endocytosis, and are converted back to ACH through recombination with acetyl groups. Neonicotinoids are synthetic molecules that can bind to acetylcholine receptors in insects. However, they cannot be broken down by acetylcholine stearase, and thus don't unbind from receptors. This blocks synaptic transmission, leading to paralysis and death.